So continuing, meiosis has two cell divisions. They are divided into the same phases that we had for mitosis. However, they are called one or two depending on which part they are. Note that if you do not label something, let's say you just said interphase, you would be indicating that that is mitosis, not meiosis. So in this case, if you have interphase 1, that's different than interphase 2. So you're going to see that there's some differences. By the way, interphase 2 in some books and in some references that you will see is sometimes skipped because it's very, very short. There's not a whole lot that is happening there, but I'll talk about what is happening there. That some people label interphase 2 and some people do not. So we'll just kind of go through the process. Now this handout right here is one that I gave you. So right the second, stop the video and get it out. Okay, so we're going to talk about each of these parts. What I've drawn here for each part is I've drawn a cell that starts out with two homologous sets. So this is a diploid cell. And I've just drawn one big, one small homologous set. So this would have a diploid number of 4, or that is 2n equals 4, where n is the number of homologous chromosomes. All right, so in this case, what's going to happen is that I do G1, which is, remember, the growth of the cell getting ready to divide. Then I do synthesis, which is the replication of chromosomes. And then I do G2. What is happening in G2 really is that the centrioles are dividing and the microtubules are assembling. So this is all written out for you when we did mitosis, so I'm not going to write it out again. What I've just written on this handout is what's kind of different between meiosis and mitosis, okay? All right, so then what happens is prophase 1. So in prophase 1, of course, in the early part of it, the chromosomes are coiling up. The spindle is starting to form between separating centrioles. And, of course, there's the aster on the end. But what's really different is that instead of individual chromosomes being free in the cytoplasm at the end, the tetrads that we just talked about and crossing over that we just talked about is occurring. So the tetrads are going to be joined to the spindle. So you have the homologous pairs paired up with their sister chromatids, and they will be crossing over at this point or exchanging information. Now you won't really see this crossing over until anaphase. What happens in metaphase is that the tetrads line up. So instead of having 4 individual chromosomes lining up like you would in mitosis, we have the homologous sets together. And then in anaphase is when they pull apart. So this is when the cell actually goes from diploid to haploid. This is called reduction division. And by the end of telophase, you will have a completely reduced chromosome. So in telophase 1, all the cells are 1n. Okay, so in anaphase, you can see the results of crossing over in this diagram where the homologous sets of chromosomes have exchanged information indicated by the light and dark parts. So in anaphase 1, what happens, of course, is the tetrads separate, and now the chromosomes move towards the poles. They are called dyads. They're called dyads just to indicate that they are not sister chromatids that look exactly alike, but basically they're kind of like the sister chromatids from one homolog separating to the other side, separating to separate sides, Okay, so at the end of telophase 1, then you have haploid cells. However, the sister chromatids of each of the chromosomes is still attached, or these are dyads. And then you enter into meiosis 2. Now, meiosis 2, as stated before, we don't really have much of a G1. It's very, very short. That is, the cell may recoup some energy loss, like make more ATP, for example, or something like that. But basically, there's not much G1. There certainly is no synthesis because the cell does not replicate chromosomes again because it already did it the first time. And so what really is just happening is G2. G2 is where the centrioles divide and make 2 of them for each cell. This is going to be for creating the spindle. So that's the main thing that happens. And like I said, some books don't even consider that it has has an interphase at all, interphase 2. Some people just call it interkinesis, all right? So then in prophase 2, what is happening is the spindle, early prophase 2, the spindle is forming between the separating centrioles. The chromosomes, of course, are coiled, and then they are going to be free and attached to the spindle. This looks a lot more like mitosis, except for we have half the number of chromosomes. So in late prophase 2, you have individual chromosomes attached to the spindle. In metaphase 2, you have the chromosomes lining up, or the dyads lining up along the poles. And then in anaphase 2, you have the dyads separating. That is, the sister chromatids of each one are separating to separate sides. But notice, because of crossing over, that they each are different. So the dyads separate and they become chromosomes again as they move towards the poles. And then, of course, at the end of telophase 2, cytokinesis would be complete, the spindle completely gone, nucleus and nucleolus reformed, and only one centriole per cell. So notice that each cell is different. These are 4 haploid cells, and that is 1N. That is, if N was the number of original homologous sets, that is still 2. So it would be 1 times 2, or 2 cells per each. Started with 4, ended with 2. All right? Now each of these cells would either be sperm, or they would be egg or ovum in humans or in other organisms. But these are what they call the gametes.